Good evening. We stand at the precipice of the microscopic empire, preparing to descend from the expansive, bounding efficiency of the macrofauna to the meticulous, invisible empire beneath our feet, and within the dense shadow of the forest canopy, the Hymenopteran order, the Formicidae. Tonight, we undertake a comprehensive, comparative analysis of 3 species of ant, each an architect and engineer of its distinct microbiome: the black garden ant, Lasius niger; the yellow meadow ant, Lasius flavus; and the southern wood ant, Formica rufa. This is not merely an observational study. This is an exploration of eusocial life as a divergent evolutionary strategy. These organisms are complex, self-organizing superorganisms, obligate practitioners of a collective existence. By examining these 3 contemporaries, we unveil the divergent evolutionary strategies that allowed the same foundational template, the ant, to dominate the soil, the pasture, and the timberlands of the Palearctic and Nearctic realms. This is a study in niche partitioning, chemical ecology, and the staggering thermodynamic austerity required to build and maintain an empire 1 grain of sand at a time. Our journey must first begin by examining the genetic contract itself, the fundamental biological architecture that permits its very existence. The evolutionary jump to eusociality, the true, complex social structure, defined by overlapping generations, cooperative brood care, and a reproductive division of labor, is underpinned by the unique haplodiploidy sex determination system. This is the ultimate engine of cooperation, providing the indispensable mathematical incentive for the worker caste to forego personal reproduction in favor of colony maintenance and the raising of sisters. We must understand the mechanics of this asymmetric relatedness. In the Hymenoptera, gender is determined by the fertilization status of the egg. The gynomorpha and fertile females store sperm from their mating flights in a specialized organ called the spermatheca. A queen determines the sex of her offspring at the point of oviposition. Fertilized eggs, which are diploid, develop into females, either workers or queens, and unfertilized eggs, which are haploid, develop into males, the drones. This system creates a profound genetic asymmetry. A female worker shares 100% of her father's genes, since the father is haploid and gives all his genes and 50% of her mother's genes. Consequently, a worker ant is more closely related to her full sisters than she would be to her own offspring. This is the core tenet of the kin selection hypothesis. The decision to sacrifice one's own reproductive fitness for the colony is rational under Hamilton's rule, where the benefit to the recipient must exceed the cost to the altruist. In a haplodiploid system, the high relatedness between sisters ensures that the massive collective benefit of raising fertile future queens, her sisters, significantly outweighs the cost of worker sterility. Yet, this genetic structure is also the source of an inherent, constant conflict, often termed the sex ratio conflict. The queen, related equally to her sons and daughters, seeks to invest colony resources in a 1-to-1 male-to-female ratio in terms of biomass, not number. The workers, however, due to their higher relatedness to sisters than brothers, genetically favor a 3-to-1 female-biased sex ratio. This conflict manifests in the final stage of sexual production, where workers may selectively kill male pupae, a behavior known as male killing, to enforce their preferred female-biased allocation. Research into Lasius niger and Formica rufa colonies has empirically demonstrated that the actual sex ratio often deviates towards the worker-preferred ratio, suggesting that the distributed power of the worker majority is successful in enforcing its genetic interests, a stunning example of collective worker agency overriding the reproductive imperative of the queen. The final enforcer of the eusocial contract is the pheromonal economy. The queen continuously produces queen mandibular pheromones, or QMP, a complex, volatile chemical cocktail that signals her health and presence. This QMP, circulated via trophallaxis, the mouth-to-mouth liquid exchange, performs three critical roles. First, it chemically suppresses the ovarian development in workers, enforcing sterility. Second, it acts as the primary colony identity signal, guaranteeing all members carry the same scent. And third, it functions as a primer pheromone, modulating gene expression. In the absence of QMP, workers will rapidly develop ovaries, begin laying unfertilized male eggs, and the colony will descend into reproductive chaos. Therefore, the superorganism's stability is chemically sustained through a complex blend of cuticular hydrocarbons and glandular secretions, a continuous chemical conversation that defines who belongs and who must be expelled or killed. Having established the necessary foundation of the eusocial contract, we turn our attention now to the most ubiquitous of our triad, the black garden ant, Lasius niger. This species is a biological triumph of generalism, whose success is not built upon specialized defense or massive architecture, but upon its behavioral plasticity and its seamless, non-specialized adaptation to disturbed, urban, and agricultural environments, a trait known as commensalism. Lasius niger workers are small, typically 3 to 5 millimeters long, with a uniform dark brown or black coloration. This pigmentation provides maximal melanin-based protection against the harsh UV radiation and temperature fluctuations of exposed urban habitats. They are monomorphic, meaning there is little size variation among the worker caste, which contrasts sharply with the polymorphism seen in Formica rufa. The colony's lifespan is remarkable. While a worker lives only a few months, the monotonous queen is capable of living for up to 28 years in laboratory settings, a longevity that allows the colony to survive decades of resource fluctuation, a strategy known as polydomy of time. The architecture Lasius niger is defined by simplicity and adaptability. Nests are typically monotonous, a single structure located beneath pavement slabs, stones, or wooden planks. The use of stone and asphalt for cover is a strategy of passive solar heating. These materials absorb solar radiation, transferring heat to the shallow nest chambers directly beneath them. This allows Lasius niger to maintain crucial thermal gradients, ensuring optimal temperatures, often 28 to 32 degrees Celsius, for the fastest development of brood. Unlike the deep, thermally buffered nest of Lasius flavus, the Lasius niger nest is highly susceptible to external temperature shifts, necessitating constant worker vigilance and brood shifting, moving pupae and larvae between chambers to track the optimal temperature zone throughout the day. The galleries themselves are shallow, reflecting the ant's reliance on surface foraging and its need to easily access food sources above ground. This shallow, dynamic nest contrasts sharply with the deep, permanent fortress of the wood ant. Lasius niger is a master of collective foraging. When a scout locates a rich food source, she initiates recruitment through a precise, highly volatile chemical vocabulary. The primary compound responsible for laying down the food trail is a volatile short-chain hydrocarbon mixture deposited from the Dufour's gland located near the abdominal tip. These trails are extremely quick-acting but also highly dissipative. They evaporate quickly. This volatility is a key adaptation. It allows the colony to rapidly switch focus when a resource is depleted, preventing workers from wasting time on old, barren trails. The ant's foraging is therefore characterized by rapid, ephemeral recruitment. The economic model of Lasius niger is defined by its relationship with aphids, a mutualistic partnership known as trophobiosis, or ant farming. They specialize in stem and leaf aphids, which feed on the phloem of plants. The ants consume the honeydew, the sugary excretion of the aphid, which provides the colony's essential carbohydrate supply. The workers actively patrol their aphid herds, aggressively deterring natural aphid predators like ladybird beetles and hoverfly larvae, demonstrating a complex system of livestock protection. A remarkable example of Lasius niger's husbandry is its overwintering strategy. Before winter sets in, workers will actively carry aphid eggs, and sometimes live adult aphids, down into the deepest, thermally stable chambers of their nest. These eggs and adults are protected until spring, at which point they are returned to host plants to restart the honeydew production cycle. This management reflects an advanced, multi-seasonal, and highly organized agricultural system. Nuptial flights, highly synchronized, massive events, usually occur during the warm, late summer months, often resulting in widespread establishment of new solitary colonies. In essence, Lasius niger is a high-risk, high-reward opportunist, fast, flexible, and perfectly adapted to the chaotic, fragmenting environments created by human civilization. We now transition, in a profound study of evolutionary divergence, to the life of the yellow meadow ant, Lasius flavus, a species that represents the complete antithesis of its black cousin. It is the subterranean pastoralist, a creature utterly subordinate to the deep, stable soil of grasslands. Lasius flavus is the ultimate specialist and the premier practitioner of obligate trophobiosis. The most visually striking feature is its pale yellow-orange hue and delicate structure. This is not accidental, but an explicit marker of hypergeal adaptation, life underground. The absence of dark melanin is a result of minimal UV exposure. This physiological shift permits significant energy savings, as melanin production is metabolically costly. Crucially, the worker caste exhibits significant sensory reduction. The eyes of Lasius flavus workers are dramatically reduced in size and complexity, often possessing fewer than 10 ommatidia, the individual units of a compound eye, rendering them functionally blind in the surface world. Their sensory world is dominated by chemosensory and vibrational inputs, using their antennae to navigate and communicate within the darkness of the soil matrix. The architecture of the Lasius flavus nest is a testament to thermal and hydraulic engineering in a stable environment. While the main chambers are deep in the soil, the most visible feature of its empire is the ant mound, a distinct, low dome of finely sifted soil particles built on top of the subterranean nest structure. The mound's primary function is as a thermal buffer and solar energy collector. The fine, loose soil traps pockets of air, creating superb insulation against the rapid temperature swings of the surface. On sunny days, the dark base of the mound absorbs solar radiation, transferring heat to the shallow nursery chambers within the dome, facilitating brood development in a process similar to a passive solar greenhouse. Furthermore, the mound's structure also controls moisture. The conical shape sheds rainfall, preventing waterlogging in the main nest chambers below. This allows the colony to precisely regulate the humidity, a critical parameter for fungal control and brood development, deep in the core. Unlike the mobile single-queen nests of Lasius niger, Lasius flavus colonies are intensely polygynous, hosting multiple queens, sometimes hundreds, and frequently polygamous, consisting of interconnected satellite nests. This structure provides redundancy and stability in an ecologically stable, yet resource-constrained, subterranean world. The economic model of Lasius flavus is one of extreme conservatism and obligate specialization. The colony dedicates almost zero energy to external foraging. Instead, its resource base is the subterranean conveyor, an intensive farm of root aphids which feed directly on the xylem and phloem of plant roots. Workers excavate intricate chambers directly around the host roots. They do not just collect honeydew. They actively maintain the root environment, transporting the aphids to optimal feeding routes and protecting them from soil predators like springtails and mites. The entire system is built on an incredibly low energy budget. The workers are not required to travel long distances, risk predation on the surface, or engage in high-speed trail recruitment. Maximum energy is invested in maintaining the stability and productivity of the subterranean farm, leading to highly predictable, low-variance resource returns. This stability allows the massive colony size that Lasius flavus often achieves. The life of Lasius flavus is a study in specialized efficiency, blind, pale, and dedicated to the unhurried, continuous harvest of the dark underworld. From the darkness of the soil, we now ascend to the shadow of the canopy to examine the true titan of the European forest floor, the southern wood ant, Formica rufa. This species, with its colossal architecture and unparalleled chemical defenses, is the ultimate forest engineer, imposing its will upon the biome through sheer scale and a potent chemical arsenal. Formica rufa workers are instantly recognizable by their red-brown bodies and black abdomens, and their large size, up to 10 millimeters for major workers. Unlike the monomorphic Lasius species, Formica rufa is polymorphic, exhibiting a wide size variation among the worker caste. This polymorphism dictates the division of labor. Smaller workers tend to remain as nurses and interior builders within the nest, while larger, more robust workers serve as foragers and soldiers. This system maximizes the efficiency of the colony by creating specialized tools for specialized tasks. The colonies are intensely polygamous and highly polygynous, with large colonies possessing dozens of satellite nests interconnected by subterranean tunnels and surface trails. This decentralized structure provides vast defensive perimeter control and allows the colony to exploit resources over a huge territory. The Formica rufa nest structure is arguably one of the most magnificent examples of non-human architecture. The massive conical thatch cone. This is not simply a pile of debris. It is a masterpiece of bioconstruction and environmental control. The cone is composed of millions of fragments of detritus, pine needles, small twigs, resin chips, and soil. The layers are interlocked, providing excellent hydrophobic protection against rain, while also acting as a profound insulator. The scale of the colony, which can exceed 1 million individuals, allows for a phenomenon known as internal metabolic heat generation. The constant metabolic activity of the worker population and developing brood generates sufficient heat to keep the deep core chambers stable and warm, even when external temperatures plummet below freezing. Workers actively transport larvae and pupae to specific thermal zones within the cone to optimize growth rates, acting as live thermostats. Furthermore, the outer layer often incorporates resin chips. These chips, besides being structural components, release volatile compounds that act as an antiseptic, inhibiting the growth of pathogenic fungi and bacteria within the moist, dense nest environment. The colony is literally self-medicating and sanitizing its own structure. Formica rufa employs a dual strategy foraging model, making it a powerful ecological force in the forest. Firstly, in a strategy of predation and pest control, a significant portion of its diet is comprised of live arthropod prey, making F. rufa a fierce predator. They hunt ground-dwelling insects and larvae, and their mass raids on defoliating caterpillars make them keystone species in forest health, a fact leveraged by forestry management to establish these colonies as natural biological control agents. Secondly, in arboreal trophobiosis, Formica rufa also farms aphids, but primarily high in a canopy of coniferous trees. The workers maintain highly defined, cleared foraging lanes, highways in the undergrowth, to allow for efficient, high-volume transportation of both prey and honeydew back to the central nest. The use of these fixed routes minimizes energy expenditure over long foraging distances. The defense mechanism of Formica rufa is its evolutionary calling card: chemical warfare via the production and deployment of formic acid. Lacking a functional stinger, the wood ant uses its acidopore, a specialized nozzle-like structure at the tip of the abdomen, to spray a concentrated volatile stream of the acid up to a meter away toward any threat. The acid is generated in the poison gland and stored under high pressure. When the nest is disturbed, thousands of workers stream out and spray simultaneously, creating an acidic fog around the nest. This fog disorients and chemically burns invertebrates and serves as a significant deterrent to mammals and birds. The acid itself also acts as a potent alarm pheromone. As the formic acid evaporates, its volatile components signal to nestmates that an attack is underway, recruiting a massive number of defenders to the precise point of disturbance. This defense mechanism is not only a weapon, but a sophisticated, fast-acting chemical communication system for collective defense. By placing these three species side by side, we can now undertake an advanced comparative synthesis, mapping their divergent evolutionary pathways to success by analyzing their distinct energy budgets, population dynamics, and resilience strategies. The three species represent three distinct, yet highly successful, models for maximizing fitness, each governed by a constrained energy budget. The black garden ant, Lasius niger, operates on a principle of rapid reaction and high mobility. It is the ultimate generalist of disturbed urban and pavement biomes. Its foraging is opportunistic and surface-based, relying on active search for honeydew, specifically from surface aphids on stems and leaves. Its architecture is defined by simple, shallow galleries under stones that use passive solar heating. Its defense is limited to biting and the release of simple mandibular alarm pheromones. It accepts highly volatile resource returns and compensates with an extremely low barrier to colony establishment, being monotonous and rapid foraging recruitment. Its workers have a relatively high walking speed to maximize resource discovery in disturbed landscapes. The key energy strategy for Lasius niger is flexibility. Conversely, the yellow meadow ant, Lasius flavus, is a deep hypogeal specialist found in grasslands and meadows. It minimizes all energy expenditure related to surface movement and threat perception. It runs on a budget of extreme metabolic conservatism based on highly predictable, low-variance resource returns from its obligate subterranean trophobiosis, acting as a passive farm, extracting honeydew solely from root aphids deep in the soil. Its nest is an exquisitely engineered mound of fine soil, designed for precision thermal and hydraulic control. Due to its deep location, its defense is minimal, relying on retreat to deep soil and reflecting its inherently low aggression. Its slow growth rate and deep thermal buffering allow it to endure long periods of resource scarcity without high-risk external foraging. The key energy strategy for Lasius flavus is conservation. The southern wood ant, Formica rufa, on the other hand, stands apart as an arboreal and structure specialist, dominating coniferous and mixed forests. It has the highest metabolic cost per colony, necessary to sustain its massive structure and potent defense. It employs a powerful predatory, arboreal trophobiosis strategy, executing a high-volume search for both prey and honeydew from canopy aphids on conifer needles. Its architecture is the massive thatch cone, a feat of active thermal management and hydrophobic defense. Its defining defense mechanism is the ferocious formic acid spraying, a form of aerosol chemical warfare. This requires high energy throughput via its dual foraging strategy. Its workers, while large, demonstrate remarkable endurance on their foraging trails, prioritizing scale and overwhelming force over individual worker speed. The key energy strategy for Formica rufa is throughput. These reproductive strategies are equally divergent, reflecting the ecological stability of their niches. The monotonous tendency of Lasius niger, with typical colony sizes between 10,000 and 50,000, maximizes the worker's genetic relentlessness, the haplodiploidy ideal. Lasius flavus, however, embraces high polygyny and polydomy to achieve massive colony sizes, often between 100,000 and 1 million, trading high genetic relatedness for the maximization of structural stability and redundancy. The southern wood ant, Formica rufa, uses polygyny and polydomy to maintain 500,000 to 2 million workers, trading a medium degree of relatedness for defensive scale and territorial control. This suggests that the evolutionary pressure of ecological dominance and defense can override the foundational genetic incentives of kin selection, embodying the eusocial pragmatism model. The chemical communication of these three genera further highlights their ecological differences. Lasius niger uses short-chain, volatile pheromones for trail recruitment, reflecting its need for rapid temporary communication in a chaotic environment. Its alarm signals are similarly volatile, designed to dissipate quickly once the minor threat has passed. Lasius flavus relies heavily on contact pheromones, the GGS, and vibrational signals within the stable, dark nest. Trail pheromones are less essential, replaced by simple memory maps of the root systems. Its defensive chemistry is negligible, reflecting its isolation. Formica rufa's signature chemical is formic acid, a complex weapon with a dual function as a potent, fast-acting alarm, pheromone. This demonstrates the integration of chemical offense defense, and communication into a single, highly specialized system. The triad's ecological impact extends far beyond simple population figures. They are keystone species whose presence dictates soil health, hydrology, and pest pressure across vast territories. Let us consider the immense ecosystem services provided by this triad, demonstrating their role as ecosystem engineers. The constant excavation of galleries by both subterranean species, particularly Lasius flavus, increases soil porosity, a service known as soil aeration and hydrology. This turnover improves water infiltration, reduces runoff, and helps integrate organic matter into the mineral soil layers, a vast, uncompensated labor force for pedogenesis. Lasius species are also significant contributors to myrmecockery, ant-mediated seed dispersal. Workers collect seeds that possess an elaiosome, a fatty, lipid-rich external structure, consume the nutritious elaiosome, and discard the viable seed in abandoned nest chambers or refuse piles. This often results in the seed being buried in a nutrient-rich protected microclimate, significantly boosting germination success. Most notably, the colossal systematic consumption of forest insects by F. rufa serves as a primary natural pest control mechanism. A single large wood ant colony can consume millions of harmful larvae and adult pests in a single season. The health and abundance of F. rufa colonies are thus used as an indicator species for the stability and health of forest ecosystems. A collapse in F. rufa populations often precedes or accompanies a surge in herbivorous insect pests. Considering the intensifying challenge of climate change, these species show unique vulnerabilities and resistances. Lasius niger demonstrates high resilience. Its flexibility and reliance on passive solar heating allow it to rapidly relocate nests to track optimal thermal regimes in disturbed, heat-absorbing urban environments. Its generalist diet provides resilience against the failure of a single food source. Lasius flavus, however, has moderate to low resilience. Its specialization on deep, stable soil environments makes it highly vulnerable to increased drought conditions, which can lead to the death of its root-aphid livestock and the collapse of its hydraulic architecture. Formica rufa shows moderate resilience. Its vast scale and metabolic heat granted excellent protection against winter extremes, but its reliance on arboreal trophobiosis makes it highly vulnerable to forest fire severity and changes in conifer health. Furthermore, increased spring temperatures can lead to premature metabolic acceleration, causing resource shortages before the forest canopy fully recovers. The modern ecological context thus places the greatest adaptive pressure on the specialists, Lasius, flavus, and Formica rufa, while rewarding the generalist flexibility of Lasius niger. Our comparative analysis concludes with a look forward, examining how these three species serve as models for cutting-edge myrmecological and bio-inspired research. The true scientific value of this triad lies in the concept of the superorganism. The colonies are not groups of individuals. They are functional, self-regulating biological entities exhibiting emergent intelligence far beyond the capacity of any single worker. Researchers are intensely studying the foraging models. The Lasius niger algorithm, with its volatile pheromone trail system, is a model for decentralized, dynamic routing. The rapid dissipation of the signal allows the swarm to forget old solutions quickly, enabling instantaneous adaptation to changing resource landscapes, a crucial design principle for swarm robotics, operating in volatile environments. Similarly, the Formica rufa traffic control, the way wood ant workers manage high-volume, two-way traffic on their foraging lanes without congestion or traffic control, demonstrates an optimal solution for decentralized self-organization in queues, governed solely by contact and chemical cues. The future of ant research is heavily invested in deciphering the full complexity of their chemical communication beyond simple trail and alarm signals, focusing intensely on cuticular hydrocarbon profiles, or Chi-Cs. These Chi-Cs are complex, non-volatile waxes on the ant's exoskeleton, a highly complex blend of long-chain hydrocarbons. These Chi-Cs are not just waterproofing. They are the colony's signature scent, the universal solvent for social information. The blend allows workers to differentiate between a nestmate and an intruder instantaneously. The colony chemically polices itself by checking every individual against the shared template of the queen's Chi profile, which has been distributed throughout the nest via trophallaxis. Moreover, subtle variations in Chi-C profiles also indicate an ant's current task or its physiological age. This chemical tagging allows the colony to precisely allocate labor without needing a central coordinator. The worker's job title is literally written on its skin. The application of this research involves synthesizing these Chi-C blends to create synthetic pheromones capable of disrupting pest populations. For example, misdirecting Lasius niger foragers, or enhancing beneficial species, such as attracting Formica rufa to pest-infested areas. The black garden ant, the yellow meadow ant, and the southern wood ant, though physically disparate and ecologically opposed, are united by the relentless thermodynamic efficiency of their collective life. Their eusocial contract, rooted in genetic sacrifice and maintained by a continuous, nuanced chemical conversation, is the enduring architectural foundation of the terrestrial ecosystem. Lasius niger reveals the power of dynamic adaptation. Lasius flavus champions the stability of metabolic conservatism, and Formica rufa demonstrates the ecological dominance achievable through chemical warfare and collective scale. They are the ultimate biological machines, individually simple, collectively brilliant, and relentlessly governed by the deep-seated, elegant calculus of kin selection. Thank you.